let's start chapter 3. As you can see, chapter 3 talks about the cells, the living unit. Before I start, is there anyone who doesn't know what is cell? Everyone knows what is cell. That's good. So what is cell? Tell me. Huh? The smallest structural and functional unit of any living being. If you do not have a cell, you are not considered as a living being, right? For example, do you know that the virus has cell or does not have cell? No cell. Yeah. Bacteria has cell. You and I have trillions of cells, right? And those cells that we have in our body are not the same. That's the very important understanding. So in our body, we can have so many different types of the cell. If you ask me to be exact, it's probably around 250 different types of the cells that we have. Now the question becomes based on what? So you can have different type of the cells in terms of their structure. You can have different types of the cells in terms of their function. So now let's look at some of the cells right here that we have. But before I look at that, let's look at the cell model that we have, right? We have this model of the cell, and then I also try to draw a cell right here. Okay, so what do you think? When I look at the cell right here, we can see like probably a little bit of oval shape. Usually animal cells are a little bit oval, so they have this structure called nucleus, the cell membrane, and different type of the organelles like mitochondria, lysosome, centriole, Golgi apparatus, and all these things, right? But just to let you know that not all cells are structurally similar and not all cells have the same organelle. Okay, so for example, if you think about these cells that I drew right here, and the cells model that we have, we are basically trying to make a cell which is generalized. When I say generalized, what do we mean by that? It means that we are trying to assume that this cell has everything that we have to learn about. But that doesn't mean that all the cells in your body have the same structure and the same organelles. Is it clear, everyone? Yeah, good. So now let's look at some of the cells that we have in our body. So if you look at the cells right here, the fibroblast, what do you see? The cells are actually trapped in the net-like structure, right? But if you look at the shape of the erythrocyte, what do you see? Biconcave. It looks like a bowl, right? And if you look at the skeletal muscle cell versus the smooth muscle cell, if you look at the smooth muscle cells, they are like the spindle shape and they have the tapered end. And if you look at the macrophage, what do you see? They have a lots of projections. And look at the fat cell, it looks like a big blob. Okay. What about the cells which has actually head and a tail, right? So you have your neuron, your nervous tissue cell in your brain, in the spinal cord and the nerve. You have this neuron which has a what? Head and a tail. And also sperm, which is actually helpful for the reproduction, also has a head and a tail, right? So now let's talk about a cell just for understanding and learning purpose. A generalized cell, the cell we have, the cell model we have, the cell that I drew, a generalized cell, just assuming that it has all the different parts which are important. Okay, so if you want me to actually divide the cell structure, I would say the cell has actually 3 different structural parts. Okay, first of all, in the very first day we learned that the very important function in our bodies to maintain the boundary. So we not only have the boundary what? Of our body, we also have the boundary where? In the cell. And that boundary is provided by who? Provided by the cell membrane or the plasma membrane. Okay? And inside the plasma membrane, you have this, what? What do you call this part? Nucleus. Nucleus. Do you know nucleus is a very important part of your cell? Why? Nucleus is almost like the control center of your cell. Okay? It controls pretty much all the functions in your body. So that is why nucleus is called the control center of your cell. And in between the cell membrane and the what? And the nucleus, whatever you have, these are all called what? Cytoplasm. So these are the three basic structural component of your cell: cell membrane, nucleus, and the what? Cytoplasm. So now, do you remember whenever we were talking about all the what? Chemistry in our body? What is the most abundant molecule in our body? Water. Yes, very good. So the most abundant molecule in our body is the what? Water. So what does that mean? It means that you have the what? You have the cell, and cell is actually what? Submerged in the water. Submerged in the water. So you have water outside the cell, you have water inside the cell. Whatever water you have outside the cell, we call them extracellular fluid. And the fluid you have inside the cell, you call it intracellular fluid. And then not only that, in between the cell, you have the what? Interstitial fluid. Okay, so what does it mean? You are actually surrounded by water, you are submerged in the water, your cells are actually what? Submerged in the water. So these extracellular fluid are actually different type of the fluid, like for example interstitial fluid. Okay, that could be blood plasma. Do you know what is blood plasma? What is blood plasma? Does anybody know? Nobody? Blood is what? Liquid or solid? Liquid. Okay. Is there anything solid in there in the blood? Blood has different types of the cell which are submerged in a lot of water. Mostly they have the water. So when you actually take out all these what? Cell from the blood, whatever is left is called the what? Blood plasma. Okay. Then the cerebrospinal fluid, which is inside your what? Brain and the spinal cord. Okay, that is called the what? Cerebrospinal fluid. Not only that, a lot of the cells also actually secrete like fluid. Like for example, we know that our mouth is always wet. Why? Because you are secreting, your cells are secreting what? Mucus, saliva, and then the extracellular matrix substance that acts as glue to hold cells together. So now let's start with the what? Plasma membrane. So when you ask me what is cell, the first thing probably I want you to think about is cell is basically probably a big mansion. Okay, it has so many rooms. Okay, and so many rooms have so many different function, right? And it's a big house, a big mansion with so many different rooms. Okay, so now if you think about the house itself has a what? A boundary. We call it what? Cell membrane. And this cell membrane, who remembers which molecule is your cell membrane made up of? I talked a little bit about it last week. Anybody? Lipids and protein and phospholipid. Someone is trying to say phospholipid. Why not lipid only? The reason it is not by lipid only, we know that lipid is the molecule which does not like water. And your cell is actually what? Submerged in the water. So how can this lipid will be able to survive? So to be able to survive, lipid is very, very what? Intelligent. They bound with the what? Phosphate. Okay, so they have a phosphate head and a lipid tail, and this phosphate head and the lipid tail together is making the what? Phospholipid. Was it is making phospholipid. So you have water here and then you have water here. So now lipid do not want to face the water. What they are gonna do instead? They are gonna face each other. The lipid is going to do the what? Face each other and make this phospholipid bilayer. Phospholipid bilayer. So now what I want you to understand right here, there is a very important understanding right here, which is if you have a big mansion, do you want everyone to enter into your cell or enter into your house? Everybody, you want everybody to enter your house? No, you do not want that, right? So you want to make sure that your house has a really nice boundary which does not allow people to come in like whatever way they want, right? You do not want that. Exactly like that, your cell membrane is made up of the what? Phospholipid bilayer. So that is how they are making sure that not everything can enter your cell very, very easily. Okay. So now that doesn't mean that you do not want anything or anybody to come into your house. Do you want that? You do not want everybody to come to your house. At the same time, you do not like perfectly have the proper boundary so that nobody can come inside your house, right? Do you want that? You do not want both of that, right? So that is the reason you want the specific people to come to your house. That is the reason you have the what do you have in the boundary of the house? Fence for not allowing people to come. And then what do you have to allow people to come? The door or the gate. Okay, so in the cell membrane, that is why you have the what? Different type of the proteins. What do you have? Different type of the proteins, and that protein actually act as a door or the window to be able to allow things to pass through it. Okay, then you have the phospholipid bilayer, you have the what? You also have the different type of the protein. You also have another thing which is also actually lipid. We call it cholesterol. Cholesterol is also inside your cell membrane to provide the what? Proper rigidity of the cell membrane so that they have the proper structure. Okay. Now there is these things right here. Does it remind you of any structure, specific structure that we have learned last week? Polysaccharide, like a lot of the carbohydrates together, right? So now the chain of these carbohydrates could be attached to the protein. We call it what? Glycoprotein. If they are attached to the what? Lipid, we call it glycolipid. And both of them together makes the what? Glycocalyx. What do they make? Glycocalyx. So now let's talk a little bit more about it. Let me ask you some questions. So what are the structural components of the cell membrane? First of all, they have the phospholipid bilayer. And what do they have for the rigidity? Cholesterol, very good. And what do they have as door? Proteins. And what do they have? Like they also have the carbohydrate, which is attached to the what? Carbohydrates attached to the protein, we call it glycoprotein, and the carbohydrates attached to the lipid is called the what? Glycolipid. Glycoprotein and glycolipid together is called what? Together is called what? Glycocalyx. Okay, so now let's talk about, so we already know the structure. We know that in our cell membrane we have this 75%, mostly we have the what? Mostly we have the phospholipid, and we have the 5% glycolipid, carbohydrate attached to the lipid, and then we have the 20% of the cholesterol, increases the membrane stability, as I have been talking about. So now I told you that you have what? The proteins inside your cell membrane act as door, window, things like that, so that things can pass through it. Okay. So there are different types of the protein. There are some protein which are called the integral protein. When I say integral protein, what do we mean by that? The integral protein is actually what? Passing through the what? The membrane. So they are inside the membrane. Okay. And then some proteins are peripheral, not properly inside the cell membrane, but they are kind of like hanging on top of the what? Cell membrane, and we call them peripheral protein. So the integral protein, most are transmembrane protein and function as a transport protein. Transport protein. So transportation. So the door is providing the what? Transportation. And these are called enzymes and receptors. And peripheral protein actually what? What do they do? So loosely attached to the integral protein included filaments on the intracellular surface used for the plasma membrane support, function as enzyme, motor proteins for shape change during the cell division and the muscle contraction, and they also provide the cell to cell recognition and the communication. So these are the different type of the function of the protein that you have. First of all, you have the transport protein. What is the function of the transport protein? Act as a door, right? Act as a door. And then you have the receptor protein. Receptor protein. So this is something I want you to understand right here. So when we say receptor protein, what do we mean by that? So when I talk about the receptor protein, I usually talk about a story. Like for example, you and your friend is Netflixing and chilling at home. Okay. Now someone is knocking at the door. Okay. And then your friend went to the door to see who is outside. And when the friend actually was looking at the what? Looking who's outside, he saw that there is a person he knows who's very strict. The strict uncle. Who has strict uncle? I don't know if you have it. You know what I'm talking about. That strict uncle, if your strict uncle after coming to the room see that what you were doing actually with your friend, maybe it's going to be very, very mad at you. So now that friend, instead of opening the door, what do? They come to you and tell you, see who's here outside. Your uncle is there. Do you want them to see like what we are doing here? So what they do, they will actually remove all the wine bottles from there, turn off the TV, set up some books, maybe studying, writing, all these things, pretending to do that. So what is actually happening? So the person who is standing outside the door, not actually coming inside the door, coming inside the home. Still, just by standing outside, they can change the environment of the home, right? That is called the what? Receptor. That is called the what? Receptor. So one actual example right here is the what? Insulin. Okay. So what happens to the insulin basically is, what is the function of insulin? What the insulin does? Huh? What does the insulin do? What is the function of insulin? Decrease the level of blood sugar. How does it decrease the level of blood sugar? So for example, if you have in the blood, you have a lot of glucose, lot of glucose, lot of glucose. So now your nervous system will tell your pancreas to secrete insulin. So now what insulin is going to do? Insulin is trying to decrease the level of what? Glucose in the blood. So what insulin is gonna do? So on the cell, you have some protein. We call it what? Receptor. What do we call it? Receptor. So on the receptor, the insulin is going to go and bind. Okay? Not enter into the cell. Just go and bind. Like that strict uncle. Okay? Just by binding to that, your cell will receive the signal to actually send some glucose transporter, to the what? Cell membrane. So that the glucose from here actually enter into the what? Cell, so that you can generate what? ATP. Does it make sense? That is exactly what is done by the insulin, just by attaching to the what? Insulin receptor. So now let's talk about the enzyme. Have you ever heard of the enzyme before? Do you know what does it do? What does it do? Huh? Enzyme is made up of protein. Enzyme is actually protein. Breaks things down. Anybody else wants to say anything? Yeah. So basically, what enzyme does is, I told you that the biggest chemical laboratory is your body itself, your cell itself. Lot of chemical reaction that is happening inside your body. But all these chemical reactions can speed up by the what? By the action of enzyme. So what is the function of the enzyme? Enzyme is a catalyst which speeds up the reaction, and after the reaction they remain what? They remain unchanged. So exactly like, I don't know, I usually try to give that analogy so students understand better. So it's almost like, for example, there is a shy girl and a shy boy love each other, but they cannot tell each other. So now the shy girl has a friend who's like, okay, I can help you. So that friend actually go to the boy and tell, okay, she loves you and all these things. Now they take you to the what? Dating, and the friend actually come back from the scenario. So that is exactly happening. So your enzyme is just speeding up the reaction, but not changing themselves. Okay, very important. Enzyme is very important part of your body. Okay, so now let's talk about the cell to cell recognition. Cell to cell recognition. So you have that what? You have that carbohydrate chain which is attached to the what? Protein, and make the what? Glycoprotein. Just to let you know that, for example, everyone has the specific name, right? Like for example, we have Maya. We know that with her name that she is that. Okay, like that your cell, whatever cell you have in your body, also have that specific recognition. That characteristics, that recognition characteristics, is actually the what? The pattern of the glycocalyx. That is how your cell understand this is your cell. So for example, for some reason you are putting in my body Maya's cell, what is going to happen? My immune system will understand that this is not my cell, this is someone else's cells. How they are going to be able to understand that? By looking at the pattern of the what? Glycocalyx. Okay, and then the attachment to the skeleton and the intracellular matrix. So first of all, you need to understand what is the cytoskeleton. It's almost like, if you think about your body, in your body, if you did not have a bone, what is going to happen? What would you look like? Would you have a structure? You are just gonna be a blob, right? No structure, right? So like that, you can see that your cell has a structure. Why do you have a structure in your cell? The reason for that structure is basically the presence of the what? Cytoskeleton. You have that cytoskeleton which is holding the structure by attaching themselves to the what? Protein. And then the cell to cell joining. Also, you have a big mansion, that doesn't mean that you are not trying to communicate with other neighbors. You want to communicate with people, right? So to be able to do that, also protein helps. That is called the cell to cell joining. Now I did talk about the glycocalyx. Can you guys tell me now, what is the glycocalyx? What is the glycocalyx? Nobody can tell me. Yeah, you have this carbohydrates attached to the what? Protein and the lipid, and making the what? Glycolipid, glycoprotein together is called the what? Glycocalyx. And as I told you, this glycocalyx is basically the what? Specific for your own body, right? For your own self. That is how your immune system understand which one is your cell, so that they keep them what? They do not destroy them. But if they find anybody else's cell in your body, what they're gonna do? They're gonna destroy them, right? So that is what. So to be able to understand which one is your cell is actually recognized by the pattern of the glycocalyx. However, if you have any kind of like cancer or the mutation, that function actually does not work properly. Okay, so for example, how cancer cell keep growing even though the cancer cell is what? Mutated, right? They your body is supposed to actually what? Kill them, but they do not kill them because they change so quickly, they divide so quickly and all these things. Yeah. Okay, so now let's talk about uh we have been talking only about the what? Your mansion, your big house. But what if, do you want to just stay alone, or you want to communicate with other neighbors? You want to do that. That is the reason cell also has the what? Cell junction. What do they have? Cell junction. Usually have three types of the cell junction: tight junction, desmosome, and gap junction. So the tight junction is actually, as you can see, these are the interlocking protein. So what is basically happening is, if you have two cells side by side, what you are doing, you have the protein to hold them so tight so that nothing can pass through the two cells. Okay, that is why this is called the what? Tight junction. Because of the tight junction, your bladder does not leak the urine from the urinary bladder. Okay, so the next one is called the desmosome. Desmosome is, you have two cells, and between the two cells you have the protein. You have the what? Protein, to be able to what? Hold them what? Together. Like for example, if you think about your epithelial cell, in the epithelial cells, what you have is actually the layer of the cells. The reason you have the layer of the cells, because of the desmosome, these cells are very what? Close to each other. They are attached to each other. That is why you have the what? Layer of cell. Now another one is called the gap junction. I don't know about USA, but in our country, what we used to do, like for example, in my house, my mom is cooking something yummy. Okay, they would want to actually share with the neighbor. A lot of the time that sharing could be, the houses are so close to each other that they have the window. They will say, okay, I cook this, I'm just giving it to you. something like that. So that exactly happens to our cell as well. So if you have something in the cell that they want to share, they can share by the protein channel or the channel protein called the what? Gap junction. Okay. Okay, so let's see how much you have understood and remember. What are the three main structural feature of a cell? What are the three main structural feature of a cell? You have the plasma membrane, nucleus, cytoplasm. Very good. What portion of the phospholipid molecule form the internal environment of the membrane? Internal environment of the membrane. So internal, so you have this phospholipid bilayer, internal membrane. So this is basically the what? Lipid together. So internal environment is made by the lipid. What type of the plasma membrane protein provides the means for moving material across the plasma membrane? Who moves the what? What type of plasma membrane protein provide the means for moving material across the plasma membrane? Huh? Cholesterol is not a protein, right? Cholesterol provide the rigidity. Huh? Integral protein. Yeah, the door which provide the what? Transportation of the things. Okay. So now, so far we have learned that we have our cell membrane. We understood the structure of it and we know the purpose of the cell membrane. What is the purpose of the cell membrane? You do not want everybody to enter your house, right? That is the purpose of your what? Cell membrane. However, you want to allow some specific people to come to your house, right? So what does that mean? Your cell membrane is what? Not 100% permeable, but this is what? Selectively permeable. Your cell membrane is what? Selectively permeable. They only allow specific things to enter into the cell, specific things to what? Go out of the cell. Okay, not everybody can enter into the cell, not everybody can go out of the cell however they want. It doesn't happen. It's a very controlled environment. Okay, so now let's talk about the transportation through the what? Cell membrane. So the transportation through the cell membrane can happen in two ways. One of the ways is called the passive transport, another one is called the what? Active transport. First of all, let's talk about, so when you hear the name active and the passive transport, what comes in your mind? Energy. Who would require energy? Active or passive? Active. Very good. It's very intuitive, right? So when you hear, you understand that active transport require what? Energy. And passive transport does not require what? Energy. However, the passive transport, why does the passive transport happen then? That's a very important question. Okay. Why does the passive transport happen? The reason passive transport happen is because, like for example, I use some Gucci perfume on my body and then enter into the room. So someone sitting on the bag can actually smell if I just probably put it right on my body, and then when I enter into the room, you can actually smell that. Why does that happen? You are not bringing your nose to my body to actually smell that, right? Just by sitting over there, I am here. How do you actually smell that? How do you smell that? The molecules of the perfume is, if I just put it on my body, I have the what? High concentration of the molecule on my body. Okay. Whenever I have the high concentration of the molecule on my body, but how that molecule is actually going to your nose? Because from the high concentration is moving toward the what? Low concentration. So the passive transport happen because of the what? Concentration gradient. Why does the passive transport happens? What is the driving force? Concentration gradient. So the change of the differences in the concentration in the two environment can lead to the what? Passive transport. Now this passive transport can happen without the help of anything. Like for example, the example that I gave you, I am entering into the room. From my body, the perfume molecule is also going to your nose. That is happening without the help of me actually going to your nose and actually letting you smell my body, like smell my perfume. No, it's not happening like that. It's happening passively. I don't have to do anything about it. You don't have to do anything about it. It's just happening, right? Passively, without the help of anything. So that is why this is called the what? Simple diffusion. That is why this is called the what? Simple diffusion. Exactly like if you take a bucket of water and then you put something colored, like for example, I don't know, do you understand, do you know the tang powder, which is yellow in color? If you just put some of it, after some time what's going to happen? They're going to get mixed with the all water. So that is basically the what? Passive transport. Now let's talk about facilitated diffusion. So when we say facilitated diffusion, so the diffusion is happening, so molecules are moving from the high concentration to the low concentration, but that is happening by what? The help of something, help of a protein. When that is happening by the help of a protein, we call it facilitated diffusion. Okay. So another one is called the osmosis. Before I talk about the osmosis, I want to ask you some question. Okay. So just by looking at the structure of the plasma membrane, you have water outside, you have water inside. What do you think is going to happen to the water? Do you think water can just pass through the cell membrane? Yes or no? Who thinks yes? Who thinks no? Okay, let's ask yes. Who thinks water can pass through the cell membrane very easily? Raise your hand. What about the rest? Three students said that they can, and you are saying no. Who says no? Raise your hand. What about the rest? No idea? Okay. So now let me give you some clue. So does water love the what? Does water love lipid? Lipid loves water? Okay. So now you have water outside and inside. Between that, you have the what? Lipid. So what does it mean? Water does not want to face the what? Lipid. So it cannot pass through the lipid bilayer easily, because lipid is the one which is the blocking it, so that water cannot pass through it. You have water inside the cell, you have water outside the cell, but they cannot pass through the cell membrane easily. Okay. Now let me ask you a question. Is it possible for them to be transported any other way instead of just going through by what? Through the cell membrane? Huh? By evaporation. Okay. So let me think about it. I want you to think about it. Like for example, whose name I remember. Let me see. What's your name? Huh? Caitlin. Okay. Caitlin is standing right outside of this wall. Now, if I want her to come inside the room, do you think she can come inside the room through this wall? He can. She cannot, even though she is just outside the room. But if I open this door for her, can she enter into the room? Yes. So that is exactly what is happening. That is why this is called the what? Facilitated diffusion. So water cannot pass through the lipid bilayer. However, they can pass through the what? Door. And that the door is facilitating the transportation of the water. Okay, and that door, that is why this is called the facilitated diffusion. So facilitated diffusion is facilitated by a door, and when we say door, what we are actually talking about? The protein. Very good. The protein, the transport protein. Okay. So now if the water is actually entering into the cell by the door, we call it osmosis. So whenever we are talking about the osmosis, we are only talking about the what? Transportation of the water molecule. What we are talking about? Transportation of the water molecule. So now what do you think? Is it clear to you, everyone, that how water molecules actually enter into the cell? Normally, through the cell membrane, they cannot pass through. However, they can only pass through by the what? The door. So, facilitated diffusion. Okay. Now, let me ask you some question. The simple diffusion, which does not need any help, is just happening passively. Okay. So what do you think? I told you water cannot pass through the lipid bilayer. What do you think can pass through the lipid bilayer? Proteins and lipids. Proteins and lipids, both of them are actually lipid is hydrophobic, but protein is uh, philic. So protein cannot, but lipid can. So lipid can pass through the what? Small, like simple diffusion. What else can pass through the simple diffusion? Remember, we talked about some molecule which are what? Non-polar. Non-polar. They have the non-polar covalent bond, like oxygen molecule, carbon dioxide molecule. The small molecule which is non-polar. That means they are not hydrophobic or hydrophilic. Okay? They are kind of like, they are like, okay, I can let them in. I don't have any like I do not like them, at the same time I do not hate them, so I can just let them in, right? So who can pass through the cell membrane by the simple diffusion without the help of any protein or anything? Who can pass through? Only small molecule, small non-polar molecule like oxygen, carbon dioxide, but polar molecule like water molecule cannot pass through. But also some lipid can pass through, because inside the cell membrane, what do you have? What do you have? Huh? Lipid. Okay. So now what do you think about the glucose? Glucose can pass through the cell membrane like that? No, they also need the what? Transport protein. So that is also happening. You have the high concentration here, you have the low concentration here. So from the high concentration to low concentration, it's happening only by the help of this what? Glucose transporter protein. So that is why this is what? Facilitated diffusion. But instead of water molecule, instead of glucose molecule passing from the high concentration to low concentration inside the cell, what if the water molecule is actually passing from the high concentration to low concentration without the help of the protein? What do you call that process? Osmosis. You call that process what? Osmosis. The glucose transportation is happening by the facilitated diffusion, and oxygen, carbon dioxide transportation is happening by the simple diffusion, and water molecule is transported by the what? Osmosis. Clear up to that? No question. Very important. Okay, make sure you understand that. Okay, so now we know, so this is the simple diffusion. If you put some colored substance in the water, right away they are getting mixed up. So from high concentration to low concentration, the what? The molecules are moving. That is called the simple diffusion. And usually by the simple diffusion, who can pass through? Lipid molecule, lipid soluble molecule, and some non-polar small gases. Okay, and by the facilitated diffusion, so if you have the door which opens and close to allow the molecules to pass, like glucose molecule to pass from the high concentration to low concentration, is called the what? Facilitated diffusion. Sometimes the facilitated diffusion can happen through the what? The channel as well. Okay, so when we say osmosis, what we are talking about? The transportation of who? The transportation of water molecule from the high concentration of the water to the low concentration of water. So this is very important for you to understand. Okay, so when I say high concentration of the water to low concentration of water, this is a very important understanding right here that you need to understand. So let me just draw some scenario, then you will understand. And it's a very important understanding, because I did ask a lot of question about that in your exam. So now, for example, if I am talking about your cell, okay, now this is inside the cell, and now this is outside the cell. Okay, so inside the cell you have 10% solution. Do you know how to make 10% solution? How to make 10% solution? Who knows that? Okay, first let's talk about 10% solution, and here you have 20% solution. Which one has the high concentration? Huh? Which one has the high concentration? Outside the cell, 20% is the what? High concentration. It's very easy to understand, right? So when you actually make a solution, how do you make a solution? So for example, to make the 10% solution, what you are doing? You are using 10 gram of solute plus what? 90 ml of water, right? And how do you make this one right here? 20 gram of solute, and how much water? 80 ml of water. So which one has more water? Inside the cell or outside the cell? Huh? Inside the cell you have more water? Okay. So if you have more water inside the cell, what is going to happen? Where do you think water is going to move? Water is going to move from inside to outside or outside to inside? Huh? Water is going to move from inside to outside. Very easy understanding, right? Right? Yeah. But you know what? In your exam, if I ask you that question, 50% of the students will make mistake. Now, why students make mistake? Let's talk about that. Because one thing I did not talk about, which is the transportation that is happening. The water is actually moving through a protein called, it's a channel protein. This protein is called aquaporin. What do we call it? Aquaporin. Aquaporin is the name of the protein which actually help transport the water. So in this scenario, everyone understand that water is moving from the what? Extracellular to intracellular. Everyone understand that, right? So now whenever we are talking about the osmosis, you actually have to think about some term, and the terms are called tonicity. The differences in the concentration, this is called the what? Tonicity. This is called the what? Tonicity. So now whenever we talk about the tonicity, we talk about it only, we are talking about the concentration of the outside in comparison to what? Inside. But whenever we are talking about the tonicity, we are only talking about that, about the what? Outside. First understanding. Second understanding is there are three scenario that could happen. One scenario is you have the same concentration of water inside, same concentration of water outside. What do you call it? You call it isotonic. What do you call it? Isotonic. So it's like, let me just talk about this. So this is 10%. So you have 10% also outside. So you call it what? Isotonic. So when you are placing your cell in the isotonic solution, what do you think is going to happen? Water will move from where to where? Huh? Outside to inside or inside to outside? It's going to happen both, because both of them have the same concentration. So they are going to equally getting transported from inside to outside, outside to inside. So it's not going to change the shape of the cell. Okay. Usually you want to keep your cell in the what? Isotonic solution. But when I say hypertonic, hyper means what? High concentration. High concentration outside. High concentration outside means you have the less water outside or more water outside. So high concentration outside. So now you have high concentration outside. In this scenario, what is happening? So if you place your cell in the hypertonic solution, this is the hypertonic solution. What is going to happen? Who has more water now? Huh? Outside you have more water or inside you have more water? Inside you have more water. So the water is going to move from inside to outside, and then what is going to happen? If you are moving water from inside to outside, what is going to happen? The cell is going to shrink. The cell is going to what? Shrink. Okay. So now you understand, if I ask you in your exam, if you place the cell inside the hypertonic solution, what is going to happen to the cell? The cell is going to lose water and shrink, right? Now I change it. So for example, now you are placing your cell in the hypotonic solution. So where do you have more water now? You have more water outside or you have more water inside? You have more water outside, right? The cell, the water will move from outside to inside. So if the water moves from outside to inside, what is going to happen to the cell? The cell will swell. The cell will what? Swell. Okay. And that is called, that is happening in the which solution? Hypertonic solution or hypotonic solution? Hypotonic solution. Now let me ask you one more time. If you place the cell in the isotonic solution, what will happen to the cell? No change. If you place your cell in the hypertonic solution, what will happen to the cell? The cell will shrink because they are going to lose water. If you place the cell in the hypotonic solution, what will happen to the cell? The cell will swell because they are going to receive a lot of water. Okay, so these are the questions I asked in your exam, and then let's see how much you actually remember. Okay, so now let's talk about the passive transport. You did not require any what? You did not require any energy. But in the active transport, you do require what? Energy. In the active transport, what do you require? You require the what? Energy. In the active transport, you do require the energy. So now this active transport, whenever active transport is happening, a lot of the times during the active transport, two things actually what? Pass together. If both of the molecule is coming to the same direction, we call this molecule, this protein, symporter. If that is happening like this, one is going out, one is going in, that is what is called the what? Antiporter. So for the active transport, you need the energy and you also need the what? Transporter, and we call them either symporter or antiporter. Okay. Now This active transport also can happen in two ways. One of them is called the primary active transport. Another one is called the what? Secondary active transport. So you can have the primary active transport, and then you can also have the what? Secondary active transport. So what is the difference between the primary active transport and the secondary active transport? So now let's talk about the primary active transport and the secondary active transport. For example, in this situation right here, to move something out, maybe like you are trying to take the sodium out of the cell. While you are taking sodium out of the cell, you are taking some potassium inside the cell. So while you are doing that, so a lot of the time whenever you are trying to take the sodium out of the cell, you already have more sodium concentration outside the cell. Okay, you are trying to what? You are trying to transport the molecule from low concentration to the high concentration. In the passive, you are transporting from high concentration to the low concentration, but in the active, you are moving from the what? Low concentration to the high concentration. So whenever you are moving the molecule from low concentration to high concentration, that is when you have to what? Use the ATP. Okay, so whenever you are using the ATP to move materials from low concentration to the high concentration, and also you are taking some potassium in, that is what is called the what? Primary active transport. So during the primary active transport, you are using the ATP. What? You are using the ATP directly. Now you already have a lot of sodium ion outside. You have a lot of sodium ion outside because of the help of the what? Energy. So now since you have a lot of sodium ion outside, and there are some also glucose molecule outside. Now this glucose molecule does not want to go from outside to inside whenever you have a lot of glucose molecule inside already. So you are moving things from low concentration to the high concentration. So the glucose is like, no, I do not want to go there. They already have a lot of glucose. So now you are like forcing, no, you have to go. Then the glucose molecule is like, I will only go inside the cell if I can go with my friend. So who's the friend? The friend is the sodium. They will only go inside the cell with glucose and sodium together whenever they can come together. Okay, they are all both entering inside the cell in a same direction by the same porter, but they are going inside the cell together because you already have a lot of sodium outside by the help of the what? Primary active transport. So here by the secondary active transport, you are not actually using the what? Using the ATP directly. Instead, you are using the help of the primary active transport indirectly. That is why this is called the what? Secondary active transport. So this primary active transport, the most important example is the sodium potassium pump. So this is the sodium potassium pump. So as you can see right here, the yellow is the what? The yellow is the sodium and the blue is the potassium. You can already see that you have high concentration of sodium outside. So you are trying to take sodium from the inside to the outside, where you are trying to move it from the low concentration to the high concentration. That is why you have to use the what? ATP. So by this transporter, you can actually take out three sodium molecule together, by the help of ATP. While you are actually trying to transport three sodium ion together, you are also taking two potassium ion inside the cell. Okay, and this is what is called the sodium potassium pump. Very important understanding. This is gonna come back again and again and again and again while we will be talking about the muscle and the nervous system. Because of the sodium potassium pump, you can actually maintain the membrane potential inside your cell. Okay, so the secondary active transport, as I told you, by the use of the primary active transport, you are increasing the what? Sodium ion outside. Now you are making, bringing the glucose inside by the help of the sodium, right? So you are not directly using the what? You are not directly using the what? ATP. So that is why this is called the secondary active transport. So these are not only the example of the secondary active transport. There are some other example of the active transports are called the vesicular transport. The vesicular transport. So when we say vesicular transport, the transport is happening by the help of the vesicle. So I try to talk about that in like for example, if you are trying to come, like for example, I came here to teach you from Denton to what is the name of this place, like TCC, right? I came from where to where? From Denton to TCC. So when I came from Denton to TCC, do you think I walked? No, I did not. I actually drove. To drive the car, I had to what? Use the energy. So the car is the vesicle, and whenever every time you use the car, you have to actually use the what? Energy, right? So inside your body, a lot of the transportation is actually happening by the car, which is vesicle. When we say vesicle, vesicles are something like this. The vesicle, you load up the vesicle and then you transport from one place to another place, like exactly like the car. And always car require energy. Okay, so that is why vesicular transport require energy. So this vesicular transport can happen, what? There are different ways. So three different types of the, first of all, you need to understand is the by the vesicular transport, you can take things out of the cell. We call it exocytosis. You can also take thing inside the cell, and we call it endocytosis. Exocytosis is what? You are taking things out of the cell, and endocytosis is what? You are taking thing inside the cell. So there are three types of the endocytosis. Number one is called the phagocytosis. Have you ever heard of the term phagocytosis before? Nobody? No? Phagocytosis is basically, like for example, a, a virus or bacteria is floating around. So now what your cell is trying to do? Trying to kill them. To be able to kill them, what do they have to do? First, they have to take inside the cell. After you take inside the cell, you have a specific organelle called lysosome. Lysosome have the digestive enzyme. That digestive enzyme from the lysosome will break down that what? Agent. And when you kill them by that process, that is called the what? Phagocytosis. And when we say pinocytosis, what do we mean by the pinocytosis? Pinocytosis is cellular drinking. Okay, you have the vacuole. So we already know how water transport into the cell by the aquaporin, but this is different. This is actually cellular drinking. So what happens if you think about this one as a cell? There are so much of water, so cell is very thirsty. So what they are gonna do? They are gonna make a what? Vesicle. After they are gonna make a vesicle, they're gonna take all this water together inside the cell. It's like drinking a lot of water together. Okay, that is called the what? Pinocytosis. And the receptor mediated endocytosis is what? Right here. So you have the receptor. Remember, the insulin receptor is attaching to the what? Receptor. And then what they're gonna do? They're gonna, so when we say receptor mediated endocytosis, so you are going to make a vesicle with so much of receptor. Every time we talk about the receptor, receptor is very what? Specific. Receptor is what? Very specific. So if you have the receptor for glucose, only glucose can enter into the cell. If you have the receptor for the specific molecule, like for example cholesterol, only the cholesterol can enter into the cell. So what does it mean? So when we say receptor mediated endocytosis, that is happening by the help of the receptor, which is very specific. Only specific molecule can be what? Entering into the cell. So I think that's all about the cell transportation. We did talk about the cell transportation. Exocytosis, when you are trying to take out the what molecules out of the cell. Okay, so let's see how much you remember. What is the driving force of the diffusion? Huh? What is the driving force of the diffusion? Concentration gradient. Very good. What occurs to the tonicity of a cell when placed in the hypertonic solution? What happens when you place in the hypertonic solution? The cell is going to shrink because you are gonna lose, the cell is going to lose water. So what transport process involved in the movement of sodium down its concentration gradient is used to power another substance up its gradient? So when we say different concentration, from high concentration to low concentration, the movement of the high concentration from the, sorry, the movement from the high to low is called the down the concentration. Movement from low to high is called the up the concentration. So try to understand this question a little bit more. What transport process involved in the movement of sodium ion down its gradient? Down its gradient, high concentration to low concentration, to power another substance up its gradient? Hmm? Passive transport? Huh? Active transport, but which, what type of active transport? Primary or secondary? Huh? Which one? Secondary, of course, secondary. Remember, did you forget all of this? So sodium ion is already what? By the primary active transport, you are moving the sodium ion what? Down the concentration or up the concentration? Up the concentration. So now you have a lot of sodium ion outside the cell. So now these, since you have a lot of sodium ion outside the cell, you can easily move glucose from its low concentration to the high concentration, right? So that is by the secondary active transport. Okay.